our discussion of enzyme function and regulation, we talked a bit about energy utilization in cells. We discussed energy coupling, in which an energetically favorable, spontaneous, exergonic reaction is linked in some way to an energetically unfavorable endergonic reaction or process and drives it forward. We also talked about the fact that often these endergonic reactions are coupled to the exergonic hydrolysis of ATP to yield ADP and a phosphate ion. ATP is the central energy currency utilized in all cells, and we've talked about the fact that in most cells the energy to synthesize ATP from pools of ADP and phosphate in the cell comes from the oxidation of organic compounds, things like glucose, for example. In this video, we're going to delve into the details of the metabolic reactions by which ATP is synthesized in a typical eukaryotic cell. A set of 3 pathways and processes: glycolysis, the citric acid cycle, which is also known as the Krebs cycle or tricarboxylic acid cycle. The fellow in the photo is Hans Krebs, who won the Nobel Prize in 1953 for working out the details of this important pathway, and oxidative phosphorylation. Collectively, these 3 pathways are referred to as cellular respiration. But before we look at those in detail, let's start by taking a look at how much ATP an organism like a human actually burns through in a day. Given some simple assumptions, it's surprisingly easy to estimate that and kind of fun to think about. First, some assumptions. Let's say we're looking at ATP utilization in an active adult male who consumes 2,800 food calories per day. Each food calorie is actually a kilocalorie, 1,000 chemistry calories, which, as you'll recall, is the amount of energy required to raise the temperature of 1 gram of water 1 degree Celsius. That's equal to 11,700 kilojoules of energy. Let's further assume that he's in energy balance. That is, the amount of energy he's taking in meets his body's needs with no deficit or excess. Let's also assume that on average, the efficiency of converting the energy in those food molecules to energy stored in ATP molecules is around 50%. So of the 11,700 kilojoules of energy taken in, about 5,860 kilojoules is captured and converted to energy in ATP. The rest is waste. On top of those assumptions, we know that hydrolysis of ATP to ADP and phosphate is exergonic, with a change in free energy of negative 30.5 kilojoules per mole. Therefore, the synthesis of ATP from ADP and phosphate must be endergonic and require 30.5 kilojoules per mole of ATP synthesized. We also know from its molecular formula that ATP has a molecular weight of 551 grams per mole, so we can do the math. 5,860 kilojoules of energy per day divided by 30.5 kilojoules per mole comes out to 192 moles of ATP synthesized per day. Multiply that by the molecular weight of ATP and we get 105,864 grams of ATP, or 106 kilos of ATP, which is equivalent to 233 pounds of ATP synthesized in his body per day. Imagine that. Your body weight and ATP is synthesized by your cells every single day. Since we know how many molecules are in a mole of ATP, and assuming 10 to the 14th cells in the body, that works out to about 10 million ATP molecules synthesized per cell per second. Each cell has millions of ATP molecules that are constantly being recycled, hydrolyzed, then put back together, hydrolyzed, then put back together. And the redox reactions of cellular respiration are what provide the energy input to fuel that synthesis. So let's take a look at these reactions, shall we? To understand what happens during cellular respiration, we're going to look at the process as it occurs with glucose as the starting substrate. But understand that other organic compounds can also be used, and these will also ultimately feed into this same set of pathways. Overall, what cellular respiration accomplishes is the oxidation of glucose in the presence of oxygen in controlled stepwise fashion, and the coupling of that oxidation to synthesis of ATP, with water and carbon dioxide produced as waste products. How much energy does oxidation of glucose yield? Per mole, about 2,870 kilojoules. If we compare that to the energy yield of an average fatty acid, we can see that fatty acid oxidation is more exergonic, yielding almost 10,000 kilojoules per mole of fatty acid. There are a couple of reasons for that. First, most of our fatty acids are in the range of 16 carbons in length, compared to glucose, which is a 6-carbon sugar, so fatty acids are bigger and contain more bonds and potential energy as a result. But the other reason is that if we compare the oxidation states of the carbons in glucose with the carbons in a fatty acid, what we see is that in glucose the carbons are already in a more oxidized, less energy-rich state to begin with. Remember that oxidation involves the loss of electrons, or control over electrons, and reduction results in gain of electrons, or control over electrons. Easy to remember using the mnemonic oil rig. Oxidation is loss, reduction is gain. Well, what do we mean by control over electrons? Well, compare carbon covalently bonded to a hydrogen atom with carbon bonded to an oxygen atom. In a carbon-hydrogen bond, which atom has more control over the shared electrons? Carbon is slightly more electronegative, so technically the electrons are spending more time around the carbon nucleus, but it's negligible. But what about a carbon-oxygen bond? Oxygen is more electronegative than carbon, and that bond is quite polar, so essentially carbon has less control over these shared electrons. In being bonded to a more electronegative partner atom, the carbon is in a more oxidized, less reduced state. This table compares the oxidation states of carbon as we might commonly see it in organic compounds. You won't have to calculate oxidation numbers for this class. I'm showing you this just to explain why fatty acids would have more energy to give up than sugars do, even if they were the same size. Down here at the bottom is carbon in its most reduced state, bonded only to hydrogen atoms. Carbon has lots of control over shared electrons here. Up here at the top is carbon in its most oxidized state, doubly bonded on either side to oxygens in the molecule CO2, carbon dioxide. Oxygen is attracting the shared electrons more than the carbon. Well, what does a fatty acid look like? A long hydrocarbon tail and a carboxyl group on the end. So if we look at most of the carbons in a fatty acid, they'll look something like this ethane, bonded to carbon and hydrogen, pretty reduced. What does a glucose look like? A 6-carbon sugar with one carbonyl group and hydroxyl groups bonded to the other carbons. So those carbons look more like this ethanol molecule. Less reduced, more oxidized, less energy to give up via oxidation reactions. Okay, so that makes sense. Whenever an oxidation reaction takes place, there has to be a reduction reaction as well. If something's losing electrons or control over them, something else is gaining those electrons or control over them. At the key oxidation steps of cellular respiration, what gets reduced are coenzymes, namely nicotinamide adenine dinucleotide, or NAD, and flavin adenine dinucleotide, or FAD. Here's the structure of NAD in its oxidized form on the left and its reduced state on the right. You can see that this coenzyme is a dinucleotide, 2 nucleotides joined at the phosphates. Here's the adenine nucleotide, and here's the nicotinamide nucleotide. This nicotinamide base is what gets reduced as glucose is metabolized. It essentially picks up 2 electrons and a proton. Now remember that a more reduced state is associated with more energy, so when NAD is reduced to form NADH, it's actually being raised to a higher energy state. It's holding energy. If the NADH is then oxidized back to NAD, that energy is going to be released, and the cell can capture that energy and use it to do work, in this case to synthesize ATP. We'll see in detail how that happens through the activity of the electron transport chain later on. The structure of FAD would be very similar, but in the place of this nicotinamide base, there would be a flavin base. But the idea is the same. Both NAD and FAD are coenzymes that are reduced at key oxidation steps in glucose metabolism. They both are then later oxidized, giving up that stored energy, which is used to synthesize ATP. So let's take an overview of what happens during cellular respiration in a typical eukaryotic cell. It consists of 3 pathways and processes. The first is glycolysis, which takes place in the cytoplasm of the cell. The second is the citric acid cycle, which takes place inside the mitochondria. And the third is oxidative phosphorylation, which also takes place in the mitochondria. We're going to look at each of these in more detail in a minute, but let's first get a sense in very general terms of what's happening in each of these. The term glycolysis means sugar breaking, and that's a fairly accurate description of what happens in this pathway. Over the course of 10 enzyme-catalyzed steps, a 6-carbon sugar, glucose, is oxidized and split in half to form 2 3-carbon pyruvate molecules. Linked to these oxidation reactions is reduction of 2 NAD to form NADH, and a little bit of energy is stored in 2 ATP. Under normal circumstances, what happens next is that the pyruvate molecules are transported across the mitochondrial membranes into the interior of the mitochondria. In the process, each pyruvate is oxidized and loses one carbon in the form of carbon dioxide, yielding two carbon molecules called acetyl. In the process, NAD is reduced, so through pyruvate oxidation, we get another 2 NADH for every glucose that we started with. Each 2-carbon acetyl molecule is fed into the next set of reactions called the citric acid cycle, where each one will be oxidized completely into carbon dioxide. So per glucose, we'll end up with 4 carbon dioxide molecules here, and that's the end of our glucose. As acetyl is oxidized, NAD and FAD get reduced, so we'll end up with 2 NADH and 6 FADH2 from the citric acid cycle, and a small amount of energy will be captured and stored in 2 molecules of ATP. At this point, the 6-carbon sugar is no more. It has been fully oxidized to 6 CO2 molecules, and in the process, the cell has synthesized 4 ATP net. But much of the energy yield from glucose oxidation remains in these reduced electron carriers, NADH and FADH2. And in the final process, called oxidative phosphorylation, these electron carriers are oxidized back to their low energy state by a set of electron transport proteins. And in the process, lots of ATP is synthesized, about 28 as a theoretical maximum. So per glucose that we start with, there's a theoretical maximum yield of 32 ATP produced, 2 each from glycolysis in the citric acid cycle, and 28 from oxidative phosphorylation at the end. So now that we have a general sense of what happens in cellular respiration, let's take a more detailed view, starting with glycolysis. Glycolysis consists of a set of 10 separate enzyme-catalyzed reactions happening in the cytoplasm of the cell. We're thinking about it in the context of cellular respiration in eukaryotic cells, but it should be noted that the glycolytic pathway is conserved across all domains, and some version of this metabolic process exists in all life on Earth, with only a couple of exceptions. From an evolutionary perspective, it is the most ancient of all metabolic processes. In broad overview, what happens is that glucose is oxidized and split into 2. NAD is reduced to NADH, and 2 ATP net are produced. We're not going to look at each of the 10 reactions in a whole lot of detail, but we are going to take a look at a few key steps in the process that are particularly important. Glycolysis can be broadly divided into an energy investment phase and an energy payoff phase. In the energy investment phase, 2 ATP are hydrolyzed to phosphorylate the sugar. And in the energy payoff phase, 4 ATP are produced, resulting in an energy yield of 2 net ATP per glucose. The cell expends a bit of energy, but gets that energy back and much more by the end of cellular respiration. So let's see how this happens. Here's our starting substrate, glucose, which, as we know, is a 6-carbon sugar. Here it is in its typical ring form. The first step in glycolysis is phosphorylation of glucose to yield glucose 6-phosphate by the enzyme Hexokinase, using ATP as the source of energy and phosphate. The benefit of phosphorylating the sugar is twofold. First, it makes the sugar a bit more reactive, enabling a higher energy yield later on. But secondly, addition of the phosphate changes the structure of the glucose such that it can no longer pass through the glucose transport proteins that brought it into the cell, effectively trapping the glucose molecule in the cell. The cell can either oxidize it to produce ATP or, if energy needs are low, may be stored in the form of starch for later use. But the point is the glucose now belongs to the cell and can't leave. Let's take a look at an animation that shows the mechanism by which this reaction is catalyzed by hexokinase. Here's the hexokinase enzyme, and this is the active site where the substrates ATP, in complex with a magnesium ion to stabilize the negative charge, and glucose bind. If we zoom in on the active site and look at the amino acids in that region of the protein, we see that amino acid number 205 in the hexokinase polypeptide is an aspartic acid that sits right in the active site. And this is the key amino acid that enables catalysis to take place. When the substrates bind, we see that induced fit mechanism that we talked about before, where the enzyme sort of clamps down around the substrate so that even water is excluded from the active site. In this case, that leads to a very close interaction between the aspartic acid's carboxyl functional group and the glucose molecule. And the carboxyl group essentially picks off a proton from the number 6 carbon's hydroxyl group. This activates that oxygen to attack the terminal phosphate group of ATP, which is sitting right next to it. The phosphate is transferred from ATP to the number 6 carbon of glucose, and the products released are ADP and glucose 6-phosphate. Remember our discussion of catalytic mechanisms that enzymes use to lower activation energy? Well, which mechanisms can we identify in this specific instance? Certainly proximity effects. The enzyme active site is correctly orienting the substrate so that they can closely interact with one another, but also acid-base catalysis. The aspartic acid residue acts like a base by stealing a proton from glucose. We could look at each of the enzymes that catalyze glycolytic steps and see the same sort of highly specific interactions between amino acid R groups in the active site and the substrates that leads to catalysis. But this is just one example of what that looks like. The second reaction of glycolysis just rearranges the position of the carbonyl group, essentially isomerizing glucose 6-phosphate to fructose 6-phosphate. If cellular ATP concentrations are low, then the phosphorylated fructose will be acted on by the next glycolytic enzyme. At this third step, we see another investment of ATP energy by the cell. The enzyme phosphofructokinase, or PFK, phosphorylates the sugar again, yielding fructose 1,6-bisphosphate. And this again energizes the glucose molecule. This is also the rate-limiting commitment step in glycolysis. Once this second phosphate is added, the sugar is now committed to the glycolytic pathway. Adding phosphate increases the potential energy of the sugar. The first phosphorylation step also commits the sugar to the cell, and the second phosphorylation commits the sugar to glycolysis. At the fourth step of glycolysis, fructose 1,6-bisphosphate is cleaved in two to form one molecule of dihydroxyacetone phosphate, or DHAP, and one molecule of glyceraldehyde 3-phosphate, or GAP. These two 3-carbon compounds are freely interconverted by an isomerase enzyme, but because GAP is the substrate for the next glycolytic enzyme and is constantly being siphoned off for the next reaction, this isomerization is effectively in one direction only, from DHAP to GAP. Now we're getting ready to see how energizing and splitting the sugar results in an energy payoff. In the next reaction, catalyzed by GAP dehydrogenase, GAP is simultaneously oxidized and phosphorylated to produce 1,3-bisphosphoglycerate, or 1,3-BPG, and NAD is reduced to NADH. This 1,3-BPG is a high-energy phosphorylated compound, and since hydrolysis of a phosphate from 1,3-BPG is highly exergonic, this can be coupled to the synthesis of ATP. Remember our table of free energy changes associated with phosphate hydrolysis? Here's 1,3-BPG. Hydrolysis of phosphate is associated with a negative delta G of 49.4 kilojoules per mole. Synthesis of ATP has a positive delta G of 30.5 kilojoules per mole, so hydrolyzing a phosphate from 1,3-BPG provides more than enough energy to drive that ATP synthesis, and of course the difference is lost as heat waste. This occurs in the active site of the enzyme phosphoglycerate kinase. Two substrates bind. The first is 1,3-BPG, and the second is ADP. A phosphate group is transferred from 1,3-BPG to ADP, yielding 3-phosphoglycerate, 3-PG, and ATP. Here's our first synthesis of ATP. And because it involves the direct transfer of phosphate from a higher energy phosphorylated compound to ADP in the active site of an enzyme, this mechanism of ATP synthesis is referred to as substrate-level phosphorylation. This is in contrast to oxidative phosphorylation that we'll look at in a bit. Now remember that for every glucose we started with, we got two 3-carbon gaps, so all of the reactions that we're now seeing in the energy payoff phase happen twice. So at this point, we've broken even. 2 ATP were invested, and 2 ATP have been produced. In the next step, we essentially see another isomerization that moves the phosphate from this end carbon to the middle one, which produces a second high-energy compound called PEP, or phosphoenol pyruvate. Hydrolysis of phosphate from PEP is highly exergonic and is coupled to ATP synthesis in very similar fashion to what we just saw with 1,3-BPG. This occurs in the active site of the enzyme pyruvate kinase. Phosphate is transferred from PEP to ADP, yielding pyruvate and ATP. And again, for every glucose, this happens twice. So we get 2 ATP net and 2 pyruvates. And we're done. Well, not quite, because pyruvate still holds a lot of potential energy, as do these NADH electron carriers. And we're going to harvest that energy later on. But we're done with glycolysis at least. You don't need to memorize all of the enzymes and intermediates of glycolysis, but there are some that you should be familiar with. You should know the enzymes hexokinase and phosphofructokinase and why they are important. And you should be familiar with the intermediates 1,3-BPG and PEP and why they are important. Okay, so where are we in our cellular respiration pathway? So far, glucose has been oxidized and split in half to produce 2 3-carbon pyruvate molecules. 2 NAD coenzymes were reduced to NADH, and 2 net ATP were synthesized. The next steps in cellular respiration occur in the mitochondria, where we're going to see the completion of the oxidation of pyruvate and the harvesting of energy from reduced electron carriers to synthesize more ATP. To fully understand these next steps in the process, we need to understand mitochondrial structure more fully. Remember that mitochondria are one of 3 organelles that contain 2 membranes, 2 phospholipid bilayers, not just one. So we see an outer mitochondrial membrane and a highly folded inner mitochondrial membrane, resulting in 2 distinct compartments within the mitochondrion. The compartment inside the inner membrane, which is referred to as the matrix of the mitochondrion, and the compartment between the inner and outer mitochondrial membranes, called the intermembrane space. Now, what is the advantage of having that highly infolded intermembrane structure? It turns out that the proteins involved with the final stage of cellular respiration, oxidative phosphorylation, are found embedded within the inner mitochondrial membrane bilayers. So obviously, having this extensively folded structure increases the surface area of the membrane tremendously, thereby increasing the amount of ATP synthesis that can be accomplished by oxidative phosphorylation. To complete the oxidation of pyruvate, it is transported into the matrix of the mitochondrion. Here's our pyruvate, the product of glycolysis in the cytoplasm of the cell. Here on the right is the mitochondrion, outer membrane, inner membrane, matrix. Pyruvate is transported across both mitochondrial membranes into the matrix. Once there, in an amazing and complex series of linked reactions by 3 different enzymes collectively referred to as pyruvate dehydrogenase, with 5 separate coenzymes, pyruvate is oxidized and decarboxylated. Essentially, a carboxyl group is cleaved off here in gray, which then leaves as a molecule of carbon dioxide. As pyruvate is oxidized, NAD is reduced to form NADH. The product of the reaction is a 2-carbon molecule called acetyl, which we see here on the right. This is what remains after a carboxyl group is cleaved off of pyruvate. As pyruvate is oxidized and cleaved to form acetyl, the acetyl product is simultaneously covalently linked to a coenzyme called coenzyme A, or CoA for short. Here's the structure of the acetyl-CoA molecule. Here's the little 2-carbon acetyl group up here, and all the rest of this is the coenzyme. You can see that the acetyl group is actually covalently bonded to a sulfur atom in the coenzyme, and this squiggly line is meant to represent a high energy or unstable thiol linkage. This high energy bond makes it easier for the acetyl group to enter the next step in the oxidation process, which is the citric acid cycle. The citric acid cycle consists of 8 separate enzyme-catalyzed reactions occurring in the matrix of the mitochondria that complete the oxidation of the acetyl molecules derived from pyruvate. Linked to these oxidation reactions is the reduction of the coenzymes NAD and FAD to produce NADH and FADH2. And a little bit of energy is captured and used to synthesize ATP. We're not going to go into any great detail on the 8 reactions. What you basically need to know is why is the citric acid cycle called a cycle, what happens to the acetyl molecule over the course of the cycle, and what are the energy transformations that occur during the cycle? In other words, where does the energy in acetyl go? In broad terms, what happens is that the 2-carbon acetyl group is transferred from coenzyme A to a molecule of oxaloacetate, forming citrate or citric acid, hence the name of the cycle. Citrate is isomerized to isocitrate, which is then oxidized and decarboxylated to form alpha-ketoglutarate, losing a carboxyl group as a more oxidized carbon dioxide molecule. This is linked to reduction of NAD to NADH. So if we're keeping count, we had a 4-carbon starting substrate, oxaloacetate. We added 2 carbons to it in the form of acetyl, resulting in a 6-carbon compound. And we just cleaved a carbon off. So now we're down to 5. In the next step, we see another oxidation and decarboxylation of alpha-ketoglutarate to form succinyl. And in a reaction very reminiscent of the pyruvate oxidation reaction, succinyl is simultaneously covalently attached to another molecule of coenzyme A, which activates it for the next step. The oxidation of alpha-ketoglutarate is linked to the reduction of NAD to NADH. And as you can see, since we've cleaved off another carbon, we're down to 4 carbons in succinyl. Cleaving this high-energy bond is an exergonic reaction, coupled to the synthesis of GTP from GDP and phosphate. GTP is used for energy coupling in the same way that ATP is, so essentially this is an ATP equivalent. The product of this reaction is succinate, which is then oxidized to fumarate, which undergoes a hydration reaction to form malate, which is then oxidized to regenerate the starting substrate, oxaloacetate. These oxidation reactions are linked to the reduction of FAD and NAD respectively, so more reduced coenzymes are produced in regenerating the oxaloacetate. Now, do you have to memorize all these steps? Again, no. Understand what's going on in broad overview. Acetyl is oxidized to produce 2 CO2 molecules. 1 NAD and 3 FAD are reduced, and 1 ATP equivalent is synthesized. Understand why it's called a cycle. Acetyl is covalently bonded to oxaloacetate, and following oxidation of the carbons added, oxaloacetate is eventually regenerated. Where do the acetyl carbons go? 2 carbons get added, 2 molecules of carbon dioxide leave. Where does the energy go? Well, some of it is used directly to synthesize an ATP equivalent. Some of it is stored in the form of reduced coenzymes, NADH and FADH2, and of course some is lost as heat waste. Now remember that this cycle will have to happen twice for every glucose that we started with. Okay, so now where are we in the cellular respiration process? Glucose has been completely oxidized through the reactions of glycolysis, pyruvate oxidation, and the citric acid cycle. 4 ATP net have been synthesized, and energy has been captured and stored in the form of electrons on the coenzymes NADH and FADH2. What happens next is those coenzymes are oxidized by proteins in the inner mitochondrial membrane, and the resulting energy transformations result in the synthesis of much more ATP than we've generated thus far, about 28 ATP per glucose. And this process is called oxidative phosphorylation. Again, this process takes place within the mitochondrion and more specifically involves proteins embedded in the inner mitochondrial membrane. So to get the orientation correct, here's a mitochondrion. This is the outer mitochondrial membrane, and this is the inner membrane where the proteins are found. The compartment inside the inner membrane is the matrix, and the compartment between the two membranes is the intermembrane space. There are a number of membrane proteins and membrane-associated molecules involved in oxidative phosphorylation. There are 4 large protein complexes referred to as complex 1, 2, 3, and 4. There's an electron carrier molecule that's mobile in the membrane called ubiquinone or coenzyme Q, and a peripheral membrane protein called cytochrome C, which you may remember from our discussion of protein sequence and molecular phylogeny earlier in the course. This was the amino acid sequence alignment that was compared across species. Finally, there's a large protein complex called ATP synthase, sometimes also referred to as complex 5, the function of which, as you might have guessed, is to synthesize ATP. Collectively, this is all referred to as the electron transport chain, And these are all considered electron transport chain proteins and associated molecules. In general overview, what we see happening at this final stage of cellular respiration is that the coenzymes NADH and FADH2 are oxidized back to their low energy states as NAD and FAD. The oxidation reactions are coupled to the reduction of molecular oxygen to form water and to the movement of hydrogen ions or protons from the matrix to the intermembrane space. And the potential energy of this proton gradient is then used to synthesize ATP as the hydrogen ions flow back across the membrane via ATP synthase. Let's see how that happens in a bit more detail. Complex 1 and 2 are enzymes that catalyze the oxidation of NADH and FADH2 respectively, and the reduction of coenzyme Q. Electrons are effectively transferred from NADH and FADH2 through these complexes and deposited on coenzyme Q. Complex 3 then catalyzes the oxidation of coenzyme Q and the reduction of cytochrome C. And complex 4 catalyzes the oxidation of cytochrome C and the reduction of molecular oxygen to yield water. Electrons are essentially moved from the coenzyme electron carriers NADH and FADH2 to molecular oxygen, which then picks up a couple of protons and forms water. But here's the important part. As electrons, which are negatively charged as you know, are moved through complexes 1, 3, and 4, positively charged hydrogen ions or protons are attracted to the complexes and moved through the membrane from the matrix to the intermembrane space, forming a concentration gradient of hydrogen ions across the inner mitochondrial membrane, with a higher concentration of protons here in the intermembrane space and a lower concentration of protons here in the matrix. This concentration gradient is stored energy. It's potential energy. First, just because there's more on one side than the other, but since we're talking about protons, it's also a pH gradient, with the intermembrane space being more acidic than the matrix. It's also a voltage gradient, with more positive charge in the intermembrane space than the matrix. So the energy stored in the electron carriers NADH and FADH2 has been transformed into potential energy in the form of an electrochemical gradient across the inner mitochondrial membrane. As you can imagine, there's a powerful driving force for those hydrogen ions to move back into the matrix to dissipate that gradient. But they can't move directly through the bilayer. They're charged, and the inner mitochondrial phospholipid bilayer is almost 100% impermeable to them. They only have one path through the membrane, and that's the protein complex ATP synthase. As protons flow through ATP synthase, a process called chemiosmosis, they induce a shape change in the enzyme complex that causes the enzyme to bind ADP and phosphate and form ATP. Because the concentration gradient supplies the energy required for this activity, this high concentration of hydrogen ions in the intermembrane space is also referred to as the proton motive force. Looking at the structure of the ATP synthase complex in more detail reveals how proton flow is coupled to ATP synthase. Here's the enzyme. To get your orientation, this gray bar represents the inner mitochondrial membrane in which ATP synthase is embedded. Here's the intermembrane space. So the outer membrane is not shown, but it would be up here. And down here is the interior or matrix of the mitochondria. ATP synthase consists of multiple subunits that come together to form a stationary stator, here in yellow, the catalytic active sites where ADP and phosphate bind and combine to form ATP, down here in pink, and then a rotor region with a connected axle or rod that inserts into the catalytic knob. The catalytic mechanism driving ATP synthesis is absolutely unique. It's essentially exactly like a water wheel or turbine. Protons flow down their concentration gradient through the stator channel here and bind to the rotor region. When the rotor has protons bound, it is induced to rotate in the membrane, which also spins the axle that projects into the catalytic domains. As the rotor spins, the axle spins, and forces the catalytic subunits to change shape, much like a crowbar. In changing shape, the ADP and phosphate binding sites in the catalytic subunits are forced close enough to forge a covalent bond between them, yielding ATP. I know that's a bit difficult to picture from a static 2D image, so watch the YouTube animation I posted to the lecture folder on Blackboard to get a better sense of what this mechanism looks like. It's incredible. So to recap, cellular respiration encompasses glycolysis, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. Through these pathways, glucose is oxidized to carbon dioxide via the reactions of glycolysis, pyruvate oxidation, and the citric acid cycle, and the electron carriers NAD and FAD are reduced. A small amount of ATP is synthesized in glycolysis in the citric acid cycle, but the vast majority is made here at the end via oxidative phosphorylation, in which the reduced electron carriers are oxidized and their stored energy transformed into energy in the form of a proton gradient. And that energy is used to turn the ATP synthase turbine, which results in the synthesis of up to 28 ATP molecules per glucose molecule. Through these reactions, electrons move from a state of higher potential energy in organic compounds to lower potential energy in water, providing the energy to power the processes of life. Some would say that life is nothing more than a highly interconnected set of chemical reactions. In essence, we are the sum of our biochemistry. And if that's the case, then in the words of biochemist Albert Szent-Györgyi, life is nothing but an electron looking for a place to rest. In the next topic, we'll focus on feedback regulation of glycolysis and the citric acid cycle. We'll see what happens if oxygen becomes limiting in the cell. And we'll also see how this central metabolic pathway, cellular respiration, is linked to the rest of the catabolic and anabolic reactions of the cell through intermediary metabolism.